Hello everyone. So today we're mainly going to be looking at Th2 T helper cells, and we'll also be looking at different hypersensitivities. So again, recently we just talked about type 1, so if you want to go back to that lecture, we'll have more detail, but again, we'll focus on type 2 today. So CD4 positive effector T cells specialize in different actions that allow them to control the immune system. So as a refresher, we have our TFH cells, which help B cells promote B cell proliferation and differentiation into memory or plasma B cells, and also help with class switching. Th17, which are pro-inflammatory. And then again here, we have our Th2, which promote antibody production and are involved in antiparasitic responses, or the type 2 immunity. And we have our Th1 cells, which we just talked, which have our type 1 immunity. And then we have our T regulatory cells, which have a down-regulatory role. And as we've mentioned several times, Th1, Th2, and Treg cells also produce cytokines that regulate B cell class switching. So again, here's a good summary figure if you want to take another look. We've had this in multiple lectures, so I won't stay on it too long. So we've mentioned these cells many different times, but here's a good article to check out if you want to read. CD8 T helper cell subsets. So we have our ILCs, as we talked about many times. So we have our type 1. So just let me clarify. So here we have our different types of responses. So basically, in several types of literature and in basic immunology, we can say we had a type 1 response, we had a type 2 response, or type 3. So coincidentally, they line up with the numbers, at least for most of them. That makes it kind of easy. So type 2 with ILC2 to Th2, type 1 to ILC1 and Th1, and then the exception would be type 3, ILC3, and Th17. So our type 1 response, our ILC1s, which includes NK cells, result in Th1 and macrophages, which respond to intracellular pathogens. Our type 2 response includes our ILC2s, our Th2s, and IgE. This results in eosinophils and mast cells and basophils, and the response to extracellular parasites. Our type 3 response includes ILC3, Th17 cells, and our neutrophils, which respond to extracellular bacteria and fungi. So here's an overview of the development of the role of CD4 positive T helper cells in ILC subsets. So naive CD4 T cells can differentiate into Th1, 2, 17, and our TFH and our T regulatory subsets upon T cell receptor activation depending upon the cytokine present. So as I mentioned much earlier, essentially we talked about these three different pathways, but they're actually initially stimulated through our ILCs. So we have our ILC acting first, then we have our type 1 response through certain cytokines from the ILC cells and the antigen itself and the APCs. We have our Th1 response and all the processes we talked about earlier. So as I mentioned, we'll be mainly focusing on our Th2 cells. So our Th2 and T follicular helper cells produce cytokines that support B cell class switching to IgE. Th2 T helper cells support antiparasite responses by recruiting and activating eosinophils and mast cells and promoting IgE production. And they have a key role in allergic reactions, which are our type 1 hypersensitivities, which we'll discuss in detail today. So B cells require signals from recognition of the antigen, which is our signal 1, which we talked about before, and T cell help, which is signal 2. So CD40 on the B cell interacts with CD40 ligand on the T cell. Then we have our cytokines like interferon gamma, IL-4, and IL-21, which regulate B cell activity. So we can see this summarized here. Then these signals drive B cell proliferation, differentiation into plasma cells, and control class switching. So in this case, if it's Th2, we have lots of IgE released, and this is also required for maturation into memory B cells. Then we have our antibody production, and again, our antibodies have several roles, including protecting us from infection through neutralization, opsonization, common fixation, and antibody-dependent cellular cytotoxicity. So here we can kind of see an example of those here. Again, if you want to watch our B-cell and antibody lectures. So T B-cell interactions in a T cell dependent antibody responses, we have linked recognition. So a B cell specific for the same virus, but a different epitope, also processes and presents viral peptides. So again, they need to be specific for the same antigen, but not necessarily the same epitope. So here we can see we have a viral protein in red. It's presented by a dendritic cell via class 2 MHC to a T helper cell. If the activated T helper cell recognizes the viral peptides presented by the B cell, it can efficiently help the B cell as accessory signals to promote B cell proliferation, differentiation, antibody production. This is an example of linked recognition, where T helper cells are activated by the same antigens as that recognized by B cells, even though they see different epitopes. So we have our virus-specific TFH cell, provides help to B cells to recognize a linked epitope. So T helper activity is necessary for B cell activation and the control of class switching, which we've talked about many, many times. So our antigen recognition by a TFH, this includes signals that activate B cells, like we said. We have B cell proliferation, which generates plasmablasts that form primary focus. So these B cells then divide and activate. Then we have our effector cells. So further differentiation can proceed to the germinal center, resting memory B cells, or antibody secreting plasma cells. So again, this is stimulating different kind of groups of our B cells. T cell help delivered through CD40 ligand binding to CD40 on B cells, and through cytokines such as IL-4, interferon gamma, TGF-beta, and IL-6, which you can see right here. These are produced by the T cells. This results in proliferation and differentiation of B cells and controls class switching. So again, if it's Th2, go to IgE. IL-21 activates transcription factor STAT3 in the B cell, and this enhances B cell proliferation and differentiation in memory B cells and plasma cells. So here's our STAT3. We also have the activation and other pathways discussed of our transcription factors, AP1, NF-kappa B, NFAT, etc. And we also have our cytokines, which have various targets. So T helper cells stimulate B cells through CD40 ligand and by directed delivery of cytokines. So T helper to B cell interaction, we have our CD40L, which has induction on the T helper cell, and this binds to CD40. This interaction is stabilized by adhesion molecules such as the integrin LFA1 on T cells, which binds to ICAM on the B cell. So we've seen this interaction between LFA and ICAM quite often. We also have cytoskeletal rearrangement, which we saw in our last lecture in T cells. So the Golgi apparatus will be aimed to the contact point with the B cell, and this causes the directed release of cytokines. So instead of our last example where we had directed release of granules to kill a cell, now we have directed release of cytokines to help the B cell differentiate. So these signals are delivered by certain cytokines like IL-4 and IL-21, which recognize CD40 ligand signal. Then we'll have B cell activation and clonal expansion. So next, other cytokines such as IL-6 cause B cell differentiation to plasma cells and resulting in antibody production. So T helper cells make membrane-bound and secreted molecules that activate B cells. IL-4 secretion in a polar fashion by Th2 cells to the site of contact with the B cell. So here we have interaction between all our different molecules. So the T cell receptor and the MHC are co-stimulatory of CD40 and CD40L, LFA to ICAM. Then the helper T cell reorientates its cytoskeleton in its apparatus, as you can see here. And then IL-4 is released in this scenario and is confined to the space between this B cell and the T cell. So just how important are T cells to B cells? So there's evidence from T cell independent antigens. We have this independence from our T cell independent antigens, So this lack of signaling via CD40L has an impact on class switching in memory B cells. So again, we would only, without in our thymus, in our TI1 or T cell independent B cells, we have one or two, if you remember, thymus independent, we get IgM. So we have evidence from our immunodeficiencies, for example, Xlinked hyper IgM syndrome. So there are two types of thymus independent T cell independent antigens, TI1, TI2, as I mentioned. So the characteristic of these is the fact that they mainly secrete IgM and have little or no class switching. So again, this is just recap if you want to go back. And there's poor induction of memory B cells. So you can see how, without T cells, this is all we get. And you can obviously see with no class switching and no memory, that is bad news. So our thymus, I keep saying thymus, sorry about that. Our T cell independent 1 antigens activate B lymphocytes by binding to toll-like receptors rather than B cell receptors. Our TI2 antigens have highly repetitive structures. So again, I've gone over this before, repetitive structures, and this example is LPS. And these simultaneously cross-link many B cell receptors. So, Xlinked hyper IgM syndrome. So a typical profile is recurrent bacterial infections, abnormal serum antibody levels. Serum IgG and IgA are nil, which so none to very low. Serum IgM is normal to elevated, and there's no memory B cell response to repeated immunization, which is kind of crazy. So B cells in hyper IgM syndrome, they all have surface IgM, and they're none with IgG, A, or E. And the lymphoid tissues lack germinal centers, which are the locations of extensive B cell proliferation. But usually this proliferation is a response to class switching and developing large quantities of the new antibody. So patients with hyper IgM syndrome are unable to activate B cells fully. Their lymphoid tissues lack germinal centers, as you can see in the top panel, compared to here. Unlike a normal lymph node, this shows that B cell activation by T cells is needed for germinal cell formation and several important B cell functions. So another example is a CD40L deficiency. So CD40 ligand is present on T cells from the healthy donor. This binds to CD40 and has high intensity, you can see here, high intensity fluorescence peak. So T cells from patients who are unable to bind to CD40, we see negative for CD40L expression. CD25 on T cells, which has a high affinity IL-2 receptor, are present and confirms T cells are in these patients, can be activated. So essentially it shows they still have T cells, but they don't have, so T cells can still work, but essentially they can't activate with the B cells because they don't have CD40 ligand. So we have flow cytometry analysis showing that activated T cells, in purple here, from hyper IgM patients do not express CD40 ligand. Here, T cells from 2 patients and 1 healthy donor were activated with a T cell mitogen and incubated with fluorescently labeled soluble CD40 to detect CD40 ligand, or with antibody specific for the IL-2 receptor alpha chain to detect activated T cells. So in X-linked hyper IgM syndrome, the CD40 ligand deficiency illustrates the importance of the CD40 to CD40 ligand interactions between these two cells. T cells in hyper IgM syndrome do not express CD40L. B cells are not defective, but their activity is affected by the lack of CD40 ligand on the T cells. So these B cells don't progress past the IgM production, so they never class switch or are stimulated further by the T cells. This CD40 to CD40 ligand signal is important for T cell control of B cell activation and B cell class switching and differentiation to memory B cells, which can obviously be super important. So again, this missing CD40 ligand leads to hyper IgM syndrome. This inherited defect in CD40 ligand gene causes the lack of effective T cell helper to B cells. Again, T helper cell activity is necessary for B cell activation and control of class switching, but can you have too much of a good thing? So we'll take a look at this soon. So here's a view of T cell subsets involved in B cell help. T cell help are still evolving, but Th2 are notorious for their support for B cell class switching to IgE production and roles in allergic reactions. So in certain scenarios, it's not always a good thing. So these Th2 responses can got kind of an overwhelming switch of antibodies. So we kind of have an overpowered amount of IgE, which is really common in allergic reactions. So sometimes people in allergic reactions can be considered Th2 sensitized in a sense. So they kind of will respond to this antigen with a Th2 response compared to other people that will have a Th1 response, and kind of these won't get the allergic reactions, but these people will. But yeah, we'll get to that in more detail later. So, in a positive side on Th2, they're involved in antiparasitic defense, which is a key feature. So here's an example of a whipworm, which embeds in the surface of the epithelium of the colon, leaving its posterior free in the lumen. The infection with the whipworm stimulates mucus production of the gut, as well as immune responses. So these whipworms live partially embedded in the intestinal epithelial cells. This shows a cross-section of the colon of a mouse infected with the whipworm, which has induced mucus production by goblet cells, which you guys remember from physiology are the mucus-producing cells. This mucus is visible as large droplets and vesicles inside the goblet cells, stained dark blue in the image. These Th2 cells induce mucus production to protect the epithelium through cytokine production of IL-13, and an effective type 2 response can often eliminate parasites, but not always. Oops. So Th2 cells are involved in parasite defense through varied actions. So here's essentially all the process of our Th2 cells do in a helminth or a parasitic infection. So they can produce IL-13, which induces epithelial repair and mucus. This causes increased cell turnover and movement and helps shedding of parasitized epithelial cells. Mucus prevents adherence and accelerates loss of parasite. We can have IL-13 produced by Th2 cells, which can increase smooth muscle contractility to enhance worm expulsion. So again, this is just to propel it out of the system through increased contractility. We have our Th2 cells, which can activate M2 macrophages via IL-4 and IL-13, which again can induce products of arginase 1 expressed by M2 cells, M2 macrophages, which increase smooth muscle contraction and enhance tissue remodeling and repair. We can have them which produce IL-5, which recruits eosinophils. Eosinophils produce MB9, which kills parasites. They can also mediate ADCC using parasite-specific immunoglobulin, which is IgE. You'll see eosinophils come up again. Th2 cells can drive mast cell recruitment via IL-3 and IL-9 and result in specific IgMR mast cells against Helminths. So these are IgE specific, having parasitic effects. So Th2 production of IL-4 and IL-13 promotes antiparasitic defenses, including mucus production, smooth muscle contraction, M2 macrophage activation, mast cell recruitment, and promoting B cell class switching to IgE. So now we'll see here, giveaway about our type 2 is IgE and type 1 hypersensitivity. So again, now for the kind of the bad side. So IgE-mediated reactions to antigens. So there's mast cell degranulation, which is a shared feature to all IgE reactions, and these symptoms can vary with location and antigen and reaction to it. So again, here's a bunch of IgE-mediated allergic reactions. So systemic anaphylaxis, which is obviously very bad. Acute urticaria, seasonal rhinoconjunctivitis, or also known as hay fever, asthma, and food allergy. So again, we can have very intense, potentially fatal reactions, or asthma. Well, this can have different severities, and again, certain food allergies. So obviously very common are peanuts and fish allergies, route of entry oral, and then here's our symptoms. Again, if you want to take a look, just to get a refresher, you can pause the video. So we can have sensitization. So how do these type 1 hypersensitive responses begin? Like I mentioned before, we can have sensitization, which involves class switching to IgE production after initial contact with this allergen, whatever it may be, the food, peanut, whatever. The allergen is the antigen inducing an allergic reaction. So just so we can clarify, we have something called DIR-P1. This is a protease in house of So this is a protease in house dusmate feces. So this is our antigen that we're using in this picture. It's kind of gross, but it's something we probably all have inhaled at some point. And this can damage our tight junctions within cells. So we can see here the enzyme DIR-P1 cleaves occludin in tight junctions. So here's our occludin. So this actually get through our tight junctions and enters our mucosa if we inhale it, let's say. So this DIR-P1 is then taken up by dendritic cells for antigen presentation and Th2 priming. Then these dendritic cells primes the cells in the lymph node, where our Th2 cells induce B cells switch to IgE production. Then our plasma cells travel back to the mucosa and produce DIR1 specific Ig antibodies. Again, this is usually, most allergic reactions involve a sensitization and then a secondary reaction. So this DIR-P1 enters through the epithelium, then there's uptake and processing and presentation by dendritic cells. Then we have our Th2 priming, forming our DIR-P1 specific Th cells, which induce DIR-P1 specific B cells to class switch to IgE. This IgE binds to the Fc epsilon receptors on mast cells, which are coded with IgE specific for DIR-P1. As I said, when the antigen is content again, this is when we can have our issues. This is when our mast cells degranulate. So the enzyme DIR-P1 cleaves. As I said, we have our first steps that we already talked about. We have another reaction, encounter the antigen again. DIR-P1 specific IgE binds to mast cells, and DIR-P1 triggers mast cell degranulation. IgG binds to these Fc epsilon receptors on mast cells, as we mentioned, and it also cleaves CD40 from dendritic cells. As a result, these dendritic cells make less IL-12, which means less Th1 differentiation, and we have further polarization of the Th2 responses. So essentially this response gets amplified, and we get kind of bad scenario. So this re-exposure to DIR-P1 gets bound to the IgE on mast cells. There's cross-linking of the Fc epsilon receptor, mast cell degranulation, and allergy symptoms. And then again, even though we're having these symptoms, we have the downregulation of Th1 and increase in our Th2, so we can have further exacerbation. So our mast cell degranulation releases mediators producing allergy symptoms. So this is summarized in this figure, but mast cell activation has different effects on different tissues. It involves the release of mediators from mast cell granules, which produce location-dependent allergy symptoms. So one of the key factors released is histamine. Histamine causes increased vascular permeability, smooth muscle contraction, which again, if this was in the bronchioles, such as asthma, this causes an issue. We can have leaking of fluid through the capillary endothelial layer. This can result in symptoms ranging from swelling, including edema, to hypotension and shock. We can have airway muscle constriction, like I said in asthma, and gut muscle contraction in the stomach, which can cause vomiting, and such as food allergies. Mediators released from mast cell granules, including histamine, which produce a variety of effects on target cells. So again, these are just give us different locations. So for example, in our blood vessels, we can have increased blood flow through the increased vascular permeability. Then here we can see increased fluid in tissues, causing increased flow of lymph to lymph nodes, increased cells of protein in tissues, increased effector response in tissues, and hypotension, which is bad, which can potentially lead to anaphylactic shock. So in our eyes, nasal passages, and airways, we can have that decreased airway diameter and increased mucus secretion, causing difficulty breathing. We can have congestion and blockage airways, causing wheezing, coughing, and phlegm. We can have swelling and mucus secretion in the nasal passages, and ocular itching and sneezing. In the gastrointestinal tract, again, we can have increased fluid secretion and increased peristalsis. This can cause expulsion of gastrointestinal tract contents through diarrhea or vomiting. And again, these can range from kind of mild to extremely severe. So these mediators produced on mast cells include, as we mentioned, histamine, proteases, and lipid-based mediators, which include prostaglandins, leukotrienes, and platelet activating factor, and as well as cytokines and chemokines. So here's a bunch of examples of products released in type 1 hypersensitivity. So we have enzymes, including all these, which can help remodel connective tissue matrix. Toxic mediators such as histamine and heparin, which is toxic to parasites, but can have increased vascular permeability, cause muscle contraction and autocoagulation, anticoagulation. Certain cytokines, so we have a bunch of different cytokines here. Again, we can further stimulate that Th2 cell response. Chemokines such as CCL3, which attracts monocytes, macrophages, and neutrophils, which causes further inflammation. Then we have a lipid mediator such as prostaglandin and leukotrienes, and our platelet activating factor, or PAF. And again, if you want to recap these functions, I recommend reading this again. So these mast cells and basophils can duplicate the effects of the Th2 cells and stimulate further IgE production. So again, they can cause almost a positive feedback loop. So as IgE secreted by plasma cells bind to these high-affinity Fc receptors on the mast cells, these activated mast cells provide contact and secreted signals to B cells to stimulate further IgE production. So these activated basophils also express CD40 ligand, which is needed for the B cells. And again, as you mentioned, we need this for the B cells. And then this can stimulate further production. And they also have IL-4 receptors. So an antigen binding to IgE on the mast cells and basophils leads to amplification of IgE production. So it's kind of a loop. IgE bound to the Fc epsilon receptor on mast cells and basophils results in cross-linked, which are cross-linked by the allergen, result in these activated mast cells and basophils then express CD40 ligand and secrete IL-4. These interact with CD40 and IL-4 receptor on the B cells, and this stimulates B cells to switch to IgE. So essentially, the summary here is activated by IgE, express CD40 ligand, and can express and cause more IgE to be released. So again, this can stimulate and keep going and keep doing the same thing over and over again. So one important thing to keep in mind is our type 1 hypersensitivity responses to allergen is really dependent on location. So for example, entry through the blood can result in system-wide activation of connective tissue mast cells. Entry through the skin can cause local connective tissue mast cells, causing atopic eczema. We can have entry by inhalation, which affect our mucosal mast cells, causing increased mucus and secretion of mucus secretion, and muscle constriction, causing rhinitis and asthma. And again, the dose and route of allergen entry determine the type of allergic reaction that occurs. So mast cells, there are two key locations for mast cells: connective tissue mast cells and mucosal mast cells. Responses to allergens depend on which mast cells are activated, so the route of allergen delivery matters. So again, here we have intravenous in the blood, subcutaneous for the skin, and inhalation. You can see here different types of reactions. So again, location matters. So here we'll look at ingestion. So ingested allergens enter via the gut epithelium. These are through the mucosal mast cells. This can cause vomiting and diarrhea due to the fluid outflow across the epithelium and the increased peristalsis. If food allergens spread throughout the body, spread through the blood, the effects can range from hives such as urticaria and eczema at skin or to anaphylaxis. So again, this is why certain people, some people are severely allergic compared to others. So how can these acute reactions progress to chronic conditions such as allergic asthma? So in our acute response, we have our inflammatory mediators, which cause increased mucus secretion and smooth muscle contraction, leading to airway obstruction. And there's recruitment of cells from the circulation. So this is our acute response. Compared to the chronic response, which is caused by cytokines and eosinophil production. So the acute response in allergic asthma leads to Th2-mediated chronic airway inflammation. Once sensitized, again, we need to be sensitized. The allergen inhalation, so again, at another point, these IgE are cross-linked on mast cells, and these release inflammatory mediators. This causes bronchial smooth muscle contraction and inflammatory cell influx, including eosinophils and the Th2 cells. These activated mast cells and Th2 cells secrete cytokines promoting eosinophil activation, which results in more tissue injury and recruitment of more inflammatory cells, and the cycle of chronic inflammation can cause persistent airway damage. So that's why it's kind of like this cycle of it self-perpetuates and stimulates itself. It's overreactive and then it's also chronic. So this Th2 phenotype, which I mentioned that some people are more predisposed to have a Th2 response. So essentially here we have this phenotype. It's common at certain locations of pathogen entry. So example is under the skin, in the gut and airway and mucosa. So this is kind of why we can be more predisposed for a Th2 response in these areas, essentially due to evolutionary needs to have parasitic defenses. But once a Th2 response begins, it favors further Th2 development. Like we said, that vicious cycle. So Th2 and mast cells produce IL-4, and this results in B cell class switching to IgE. But it's also important to think about with a parasite. You can see how huge this is. We need this response to be, I guess, relatively powerful. You can't just have a little response because you essentially have to kill a large macroorganism as well as prevent further infection. So Th2 and mast cells produce IL-4, which causes B cells to class switch to IgE. Mast cells amplify the process and express CD40 ligand to support B cell class switching to have further IgE. Then we have our Treg cells in these locations, which normally inhibit allergic responses to common environmental antigens. So essentially, these antigens or environmental antigens are usually not strong enough to initiate a response, as our Tregs can keep it suppressed, but sometimes people are more sensitized, or for some reason or another, they respond and develop this Th2 response. So these are some ideas why allergies happen. So there's the hygiene hypothesis, which is quite common, where our modern conditions of living, where we have less exposure to microbes, that would skew responses to Th1 rather than Th2. But for example, helminths parasites induce Th2 responses, yet are associated with less allergy prevalence, which is quite interesting. So we also have the counter-regulation hypothesis, which contact with microbes may protect us from atopy by inducing regulatory cytokine production, including IL-10, TGF-beta, which downregulate Th2 and Th1. We also have Treg cells in the mucosal immune system, which suppress responses to food and environmental antigens. So essentially, it seems that it's going to be a combination of genes. So some people are just genetic susceptibility. So a lot of people can be atopic, which is kind of the term to be they're allergic. Can be passed down, so that a lot of families will be like, the entire family will have asthma, or the parents will, and some of the kids. And also be environment. So examples, we can have early exposure to ubiquitous microorganisms, early depletion of microorganisms by repeated use of antibiotics, so that can be bad. Helminths infection can stimulate a high Th2 response, such as hepatitis A. We can also have hepatitis A virus infection, and composition of the gut microbiota, which is becoming a huge topic of research in all aspects of immunity. And some people are saying it's like essentially our first developed immune system. So how we develop that microbiota early on can kind of dictate in our entire immune system development. That's a topic for later, and you guys can look into that if you like. So this decreased exposure to common pathogens and gut colonizing microbes results in less T regulatory cell induction, and therefore increased incidence of allergic responses to environmental antigens. So essentially, as a child, if you're kept in, you've probably seen this in a lot of things. It's in TV shows and etc. But if we're not exposed to enough as kids, we essentially won't develop these tolerizing mechanisms. So essentially things that we should have been exposed to and our body recognizes as something that's normal, and we don't overreact. We are exposed to later and never been exposed to it, so it assumes it's bad, which is not the case. And yeah, we can have a bad time. And again, key here is genetic susceptibility and environment. It's not one or the other. They work in tandem. But it's a very interesting topic. So again, there's also important stuff as the timing of contact with the allergen. So again, we mentioned during our development of the mucosal immune system. So mucosal permeability or reduced barrier function due to lack of IgA or gastrointestinal damage, which can be infection related. And then again, we have the role of the gut microbiota as we discussed. It also could be hereditary factors such as Th2 bias or low regulatory cytokine production, which allows for uncontrolled activation. So again, here's a bunch of risk factors. So you're young, you have an immature mucosal immune system, early induction to solid food. So sometimes giving children solid food too early can cause an issue. So there's hereditary increases in mucosal permeability, IgA deficiencies or delayed IgA, inadequate colonization of the intestinal immune system by commensal microbiota. So apparently birth by cesarean section can increase the risk. Genetically determined bias towards Th2 environment. polymorphisms of Th2 cytokine or IgE receptor genes, impaired enteric nervous system immune alterations, such as low levels of TGF-beta, and gastrointestinal infections. So all these different things can work together with genetics and the environment at the same time, and then can increase that risk to be atopic. So it's not all hopeless. So obviously, there's many different treatments for these kind of things, and we'll take a look at these now. So we know there are many immunotherapies for allergies, and definitely there's also immunotherapy for autoimmune diseases and transplantation, anti-tumor immunotherapy, and it allows immunization against infectious diseases. So again, we'll just have a quick review. So when a lot of these things, especially for immunization, and even these ones, and again, these are immunizations to help fight infection, but also these antigen things that we're talking about come in handy when we're trying to regulate our immune system. So again, what makes a good antigen? A good antigen has a protein size with a high molecular weight proteins, which are more immunogenic. Their complex structures and particulate forms are better. What is the route of administration? The dose. What is the similarity or difference from ourselves? And the ability to interact with host MSC. So again, we have intrinsic and extrinsic factors that affect the immunogenicity of proteins, and proteins are the best antigens, although responses can be induced against carbs, nucleic acids, and lipids. So why do some antigens act as allergens? So allergens are usually delivered transmucosally at a low dose, a route that favors IgE production, and they are often very stable, low molecular weight proteins. So here we can see features of the airborne allergens that may promote priming of Th2 cells. So proteins often with carbohydrate side chains that can produce T-cell responses. There's a low dose, which favors activation of IL-4, low molecular weight, so the allergen can diffuse out of particles into the mucosa, highly soluble. They can be readily eluted from the particle. They're stable. They can survive for a while. And they contain peptides that bind to host MHC class II, which is required for T-cell priming. So here's a few approaches for type 1 hypersensitivity treatment. So we can have desensitization immunotherapy. So the route of this treatment is subcutaneous, where we gradually increase the doses of the allergen, and the goal is to aim to restore tolerance to the allergen by inducing T regulatory cells, or Th1 responses, decreasing the Th2 activity. So this subcutaneous route favors the presentation by APCs making IL-12, and therefore Th1, interferon gamma, and IgG production. So again, we talked about location. So instead of being inhaled or ingested, so this is commonly done in peanut allergies or whatever, or food allergies or bee stings, where they slowly progress the tolerance they can take to this antigen. So very, very, very low amounts as they slowly progress. And by doing it subcutaneously, we can try and stimulate the Th1 response, and then hopefully this becomes a long-term. Essentially, the body will switch its memory to this kind of Th1 memory. So next time we encounter it, it will be a Th1 response, which will lack a lot of the consequences of this Th2 response. We can also have IgE blockade, which is a passive immunotherapy, where we have anti-IgE monoclonal antibodies, which bind to the Fc region of the free IgE, and these block the binding of the Fc receptor on mast cells. And here's omalizumab, which is the example. So IgE-mediated allergic diseases can be treated by inhibiting the effector pathways that lead to symptoms, or by desensitization techniques, as we mentioned, that aimed at restoring tolerance to the pathogen. Current pharmacological approaches target symptoms. So we haven't really treated the cause of asthma or any of these symptoms really. It's a very complex pathway, and it's difficult to treat because, again, we also, on one hand, we need them. So you can't just immunosuppress people. So you kind of have to treat the symptoms. So many include antihistamines, anti-inflammatory drugs, and epinephrine, which you've seen in the EpiPens. So treatments for allergic diseases. So again, we got here mediator actions. We can affect their chronic inflammatory reactions, their Th2 response, and IgE binding. So again, we have anti-IgE antibodies. We have desensitization therapy by injections of a specific antigen to help remove the Th2 response. So to help with the chronic inflammatory reactions, we can give corticosteroids and anti-inflammatories. And to affect our mediator actions, we can give antihistamines, beta agonists, leukotriene receptor blockers, and lipoxygenase inhibitors. So again, we talked about our mast cells and stuff, basophils releasing histamines, so we can block that, for example, with antihistamines. So here's an example of our oral immunotherapy for peanut allergy. So here's a look at this paper and article if you want to take a deeper look. So in 2014, there was a clinical trial of children with peanut allergy, which reported an oral immunotherapy approach led to improved tolerance of peanuts in 80 to 9% of children, which is pretty impressive. So again, the strategy is to gradually increase the amounts of peanut tolerated. So participants are given a small dose of peanut flour with a meal in a hospital setting. The same small dose was added to their meals at home over a 2-week period. The dose was then gradually increased at 2-week intervals and over the next 6 months to reach a dose of 800 milligrams of peanut flour, which is equivalent to 5 whole peanuts. There was an increase in the amount of peanut the children could tolerate. Again, this approach is being used for walnuts, eggs, milk, and a lot of other stuff. So the problem with this research and current questions are, how long does improved tolerance last without constant allergen therapy? So if we're slowly progressing, then we stop. How quickly will it return down? Is there memory, etc.? And why is this approach effective in some individuals and not others? So that's kind of stuff like why there are individual differences and why is it not just a generalized approach? And then there's also stuff of the fact that these are very controlled studies. How realistic is this in the real world without being supervised by a doctor constantly? And is it almost worth it at that point? And like, again, there's always a risk of severe reaction. So like, you couldn't just do this at home because you have to be careful with the doses. So they'd have to have prepackaged essentially. But yeah, it's a complicated issue and it's being researched heavily. So some approaches under investigation. So by decreasing Th2 activity, we will have Th1 induction by linking allergen to unmethylated CpG, which is a bacterial DNA motif, which stimulates TLR9 ligand, and this shifts the Th1-Th2 balance, causing decreased Th2 activity. So essentially, by stimulating Th1 induction through a CpG, stimulating TLR9, we can sway the balance back to Th1. We can also have blocking of eosinophil recruitment to sites of allergic inflammation by using anti-IL-5 monoclonal antibodies. So IL-5 is massive in eosinophil recruitment and activation. So by blocking that, you can kind of essentially stop eosinophils. And this is used to block IL-5 actions or blocking a chemokine receptor, CCR3, which is eotaxin receptor. So essentially blocking the recruitment with IL-5, or we can block essentially the communication and the guidance of these eosinophils, the sites of where they would cause issues, such as the airways, by blocking the chemokines. Other possibilities include inhibiting B-cell activation by blocking CD40L, interaction CD40, or blocking IL-4. Mast cell activation by IgE could be inhibited by blocking Fc receptor for IgE on mast cells. So again, here are different things. So can we block Th2 activation of B cell to produce IgE, mast cell activation, eosinophil dependent inflammation. So again, can we inhibit IL-5, block CCR3, IL-3 receptor blockade, inhibit CD4, IL-4, and IL-13, and administration of cytokines such as interferon gamma, IL-10, IL-12, TGF-beta to stimulate that Th1 response and switch the balance. So guys, I hope you enjoyed the lecture. Th2 cells are pretty important in a lot of allergies, and you probably know someone with some sort of allergy. So if you want to talk to them about their immunology after this lecture, recommend it. Good to get people to understand kind of what's happening, and they can kind of understand, oh, I take this kind of medication, that's actually what it's doing. Just good to have people learn about the different conditions. But yeah, hope you guys enjoying it. Take care.